Good afternoon, I'm Professor G. Today we're going to explore one of the kidney's most remarkable feats, how it uses the countercurrent mechanism in the nephron to concentrate urine and conserve water, especially when every drop counts. Ever wondered how your body holds on to water when you are dehydrated? Your kidneys perform a brilliant trick to concentrate urine, thanks to a process called countercurrent multiplication. Let's break it down. Blood is delivered to the kidney by the renal artery and leaves by the renal vein. While in the kidney, blood is filtered and concentrated, with the filtrate leaving the kidney via the ureter as urine. Most of the action takes place in the cortex and medulla of the kidney, where the functional units responsible for filtering are located. These units are called nephrons, and each human kidney contains approximately 1 million nephrons. Blood enters the nephron through afferent arterioles branching from the renal artery. It first reaches the Bowman's capsule, a cup-shaped structure that houses a network of capillaries called the glomerulus. Here, hydrostatic pressure forces water and small solutes, such as electrolytes, glucose and amino acids, out of the blood and into the capsule, forming a fluid called filtrate. Larger molecules, like proteins, along with blood cells, remain in the bloodstream and exit via the efferent arteriole. The filtrate then begins its journey through the nephron. It flows first into the proximal convoluted tubule in the renal cortex, then into the loop of Henle in the medulla. It flows down the descending limb and ascends back toward the cortex via the ascending limb of the loop, before entering the distal convoluted tubule, and finally the collecting duct, which leads to the bladder. As the filtrate moves along this path, selective reabsorption and secretion of water and solutes occur, transforming the raw filtrate into urine. Meanwhile, the efferent arteriole from the glomerulus forms a network of capillaries around the nephron known as the vasa recta. These long, hairpin-shaped capillaries run parallel to the loop of Henle, descending deep into the medulla and then looping back up toward the cortex where they drain into venules. The vasa recta and loop of Henle work in close coordination to establish and maintain the countercurrent mechanism in the kidney's medulla, a system that is essential for concentrating urine. Let's take a closer look at how this mechanism works. The loop of Henle is a long, U-shaped portion of the nephron that extends into the kidney's medulla. It is surrounded by the interstitium, the space between the nephron's tubules and blood vessels containing interstitial fluid. Filtrate flows down the descending limb, around the bend, and up the ascending limb. Because fluid moves in opposite directions in these adjacent limbs, this is known as countercurrent flow, forming the basis of the countercurrent mechanism that concentrates urine. The epithelial cells lining the loop of Henle regulate its permeability to water and solutes, and their properties differ between the two limbs. The descending limb is permeable to water but not to solutes, allowing water to leave the filtrate by osmosis. In contrast, the ascending limb is impermeable to water but permeable to solutes. The ascending limb has two segments, a thin segment where salts passively diffuse out of the filtrate, and a thick segment where salts like sodium, potassium and chloride are actively transported into the interstitium. This difference, passive versus active transport, is crucial. Passive diffusion relies on existing concentration gradients, while active transport uses energy to pump solutes against their gradients. Together, these processes create and maintain a salt concentration gradient through the medulla, allowing the kidney to efficiently reabsorb water and produce concentrated urine. Let's look closer at the mechanism. The key player in generating the salt gradient in the medulla is the thick ascending limb of the loop of Henle, which is densely packed with active transport proteins. The epithelial cells lining this segment separate the tubular filtrate at the apical membrane from the interstitium at the basolateral membrane. On the apical side, the primary ion transporter is NKCC2, a symport that moves 1 sodium, 1 potassium and 2 chloride ions from the filtrate into the cell. NCCK is a secondary active transporter. It doesn't use ATP directly, but is powered by the sodium concentration gradient maintained by the sodium pump or sodium-potassium ATPase located in the basolateral membrane. The sodium-potassium pump actively transports 3 sodium ions out of the cell and brings 2 potassium ions in, lowering intracellular sodium and establishing the gradient that drives sodium entry via NKCC2. As sodium flows down its gradient, it pulls potassium and chloride along with it into the cell. To prevent potassium from accumulating inside the cell, Rom-K potassium channels on the apical membrane allow potassium to leak back into the tubular fluid. Meanwhile, ClC-Kb chloride channels on the basolateral membrane enable chloride ions to diffuse into the interstitial space, following their electrochemical gradient. The combined action of these transporters removes salt from the filtrate and deposits it into the medullary interstitium. Importantly, because this segment is impermeable to water, water remains in the tubule while sodium and chloride are pumped out. This creates a hyperosmotic environment in the renal medulla, which is essential for the kidney's ability to reabsorb water and concentrate urine. As you have just seen, active transport of sodium and chloride from the thick ascending limb of the loop of Henle creates a high salt concentration in the surrounding interstitium. In the inner medulla, this is further amplified by a high concentration of urea, making the interstitium hyperosmotic. When filtrate enters the descending limb, its osmolarity is similar to that of blood. But because the descending limb is permeable to water and impermeable to salts, water leaves the tubule by osmosis into the hyperosmotic interstitium. Its water permeability is due to rich expression of aquaporin channels. Salts remain behind, so the filtrate becomes progressively more concentrated as it descends. By the time it reaches the bend of the loop, the filtrate is at its highest osmolarity. It then enters the thin ascending limb, where the properties reverse. The epithelium is impermeable to water but permeable to salts. Sodium and chloride ions passively diffuse out of the tubule down their concentration gradients and into the interstitium. Since water cannot follow, the filtrate becomes progressively more dilute as it ascends. This passive salt diffusion reinforces the interstitial salt gradient, which is highest deep in the medulla and decreasing toward the cortex. Together, 3 processes generate and maintain this gradient: water reabsorption from the descending limb, passive salt diffusion from the thin ascending limb, and active salt transport from the thick ascending limb. This is the essence of countercurrent multiplication, where a small difference in solute transport between adjacent nephron segments is amplified into a steep vertical osmotic gradient in the renal medulla. So far, we've seen how the loop of Henle establishes a steep osmotic gradient in the renal medulla, represented here by the gradient of grey tones in the background. But this gradient is delicate. If blood flowing through the kidney were to simply wash solutes away, the system would quickly collapse. This is where the vasa recta come in. These are specialised capillaries that closely follow the loop of Henle, playing a critical role in preserving the medullary gradient through countercurrent exchange. Like all capillaries, the vasa recta have walls formed by a single layer of endothelial cells resting on a basement membrane. Solutes can move across these walls via intercellular clefts or narrow gaps between the cells. What makes the vasa recta special is that their endothelial cells are fenestrated, meaning they contain numerous small pores that allow solutes and water to pass directly through the cells. This structure makes the vasa recta highly permeable to water, electrolytes and small molecules such as glucose and urea. As blood enters and flows down the vasa recta, it encounters the hyperosmotic environment of the medullary interstitium. This high osmolarity draws water out of the blood by osmosis, while allowing solutes such as sodium, chloride and urea to diffuse into the blood. As a result, by the time the blood reaches the hairpin turn at the bottom of the loop of Henle, it has absorbed a significant amount of solute from the interstitium. If the vasa recta were to exit the medulla at this point, they would carry those solutes away, diluting the interstitial fluid and disrupting the osmotic gradient that is vital for water reabsorption. Instead, the vasa recta mirror the path of the loop of Henle and ascend back toward the cortex. Now the situation reverses. The blood in the ascending vasa recta is hyperosmotic compared to the surrounding interstitium. This drives water back into the blood and solutes back into the interstitium, restoring what was taken up earlier. This process, known as countercurrent exchange, ensures that solutes are recycled rather than lost, and that the medullary osmotic gradient established by the nephron is preserved, not washed away. We now come to the final step in the body's mechanism for conserving water: reabsorption of water in the collecting duct. As the filtrate continues along the nephron and enters the collecting duct, which passes deep through the renal medulla, it encounters an increasingly hyperosmotic interstitial environment. This gradient, established and maintained by the loop of Henle and vasa recta, drives water out of the collecting duct by osmosis, across the epithelium and into the interstitial space. From there, water is reabsorbed by the vasa recta and returned to the bloodstream. Importantly, the water permeability of the collecting duct is regulated and not constant. Movement of water across the cell membrane depends on the presence of aquaporins, a family of channel proteins expressed in specific patterns across different tissues. In the collecting duct, aquaporins 3 and 4 are constitutively expressed on the basolateral surface of principal epithelial cells. Aquaporin 2 is the regulated water channel found in the apical membrane. Its abundance there is controlled by the hormone vasopressin, also known as antidiuretic hormone or ADH. Vasopressin binds to V2 receptors on the basolateral membrane. These are G protein coupled receptors that activate the GS protein to stimulate the enzyme adenyl cyclase. This enzyme converts ATP into cyclic AMP, which then activates protein kinase A. Protein kinase A phosphorylates aquaporin II proteins located in intracellular vesicles. This modification triggers the vesicles to translocate to and fuse with the apical membrane, inserting aquaporin II channels into the surface of the cell. The more aquaporin II channels that are inserted, the greater the water permeability of the collecting duct. As a result, water is drawn from the filtrate into the epithelial cells through aquaporin II on the apical side, then exits into the interstitium through aquaporin III and IV on the basolateral side, completing the process of water reabsorption. Let's take a moment to bring everything together. The countercurrent mechanism establishes an osmotic gradient in the renal medulla. It involves the loop of Henle. The descending limb is permeable to water, not solutes, so water exits the filtrate and it becomes concentrated. The thin ascending limb is permeable to sodium and chloride, but not water, and passive salt reabsorption dilutes the filtrate. In the thick ascending limb, active transport of sodium, potassium and chloride creates a hyperosmotic interstitium. Countercurrent exchange involving the vasa recta minimises solute washout and preserves the osmotic gradient in the renal medulla. Vasopressin regulates water reabsorption in the collecting duct by binding to V2 receptors, stimulating the cyclic AMP protein kinase A signalling pathway, and inserting aquaporin II into the apical membrane. As a result, water enters cells via aquaporin II, exits to the interstitium via aquaporin III and IV, and is reabsorbed into the bloodstream. Note that without vasopressin, aquaporin II remains intracellular and the collecting duct is water impermeable, resulting in a dilute urine. Dehydration stimulates the release of vasopressin from the pituitary gland, leading to more aquaporin II insertion and greater water reabsorption. The resulting urine is highly concentrated, minimising water loss. In this way, the system integrates an osmotic gradient with hormonal control to fine-tune water balance.